Yeah, let's go ahead and kick off, please. Welcome everybody to the September meeting of the Science and Engineering Council. We're still on Zoom, obviously. We're discussing, but we haven't really, I think we're not quite there yet on the in-person meetings, but I think we will get there, just give it a little more time. If you, let's see, it's coming up on the fall. We'll be putting out our annual membership drive for renewals and new members. If you know anyone or want to ask your company to join us, please do. Briefly introduce our board members here today, and then we'll turn it over to Yvonne. I'll get myself, Tim Murphy. With we have Yvonne DeGraw, Cheryl Ebner, Joseph Stronks, Mariana Grant, Steve Skopatz, and Bob Lilly. All here from the board today. If I missed anyone, feel free to speak up. Anyway, let's go ahead. I think with that little brief intro, I'll go ahead and turn it over to you, Yvonne, for talk about upcoming meetings and kick us off for today, please.
Great, thanks, Tim. Hi, everyone. Our next meeting will be on October 12th, and we'll be sending out an email soon with information about the topic and speaker. I believe the topic will be about newly developed antibiotics, but I'm still confirming details with the speaker, so we'll let you know on that. Subtitles are available during today's talk if you enable live transcript in your Zoom window, and you can type questions into the Zoom chat or ask them, when the speaker opens it up for questions. Today, we're happy to have Dr. Norbert Reich as our speaker. He's a distinguished professor in UCSB's Department of Chemistry and Biochemistry. Dr. Reich started the drug company Epigenetics and UCSB's largest science outreach program, SciTrek. He received his BS in biochemistry from UC Berkeley and his PhD in medicinal chemistry from UC San Francisco. He's an active bike rider, sailor, and consumer of good wine. He lives with his wife, 2 pugs, a cat, and an Amazon parrot. He's speaking to us today on how to use the software that controls cellular differentiation. So welcome, Norbert.
Great.
Dr. Reich.
Yes, no, that's okay.
Yes.
Thanks. Thanks for the intro, and thanks for inviting me. And I hope I make this palatable and approachable for you guys. It's always a challenge. I was just interviewed for the same basic topic by the local radio station. And so I think the last line that person gave me was, well, you made it understandable. So I'm hoping to make it understandable for you guys as well. Although I didn't have any slides, and slides can be pretty intimidating for a lot of people who are not experienced with it. So let's see if I can get through this. Okay, so now I can share, right? Yep. There we go. Okay, can you see my screen?
Yes.
Okay, great. Okay. So I forgot that title I gave, which is it's pretty good. It's not incorrect. It's just that I forget when I gave that title.
If you're interested, we'll see.
This is based. What I'm going to present to you is the front part of it is quite a bit of the biology behind what it is that we study. And then I'm not a biologist. I'm a biochemist, and we make molecules, etc. And so it'll then go over into a paper that we just published in the Journal of Medicinal Chemistry, and which highlights the 2 compounds that you see in front of you there. And they are basically, and this gets kind of technical very quick. They're in the world of drugs, if you have a new mode of action, that's a big deal because that's patentable and it's distinct. And these molecules, the 2 that you see right here, they have a completely different mechanism of action than anything that has been studied before that is used to regulate or to control these enzymes. And so that's a big part of why we were super excited about it. And I'm going through the process right now because people have told me I should. I'm going through the patenting process on this right now. Okay, so here we go. Let's see. So the field that this is part of, let me see if I can stop that there. Yeah, this every science endeavor has an area, a field that it's in. This is this field is called epigenetics, and there's a lot of things on my screen that's blocking stuff, so I can't always read stuff. And just stop that. And then for some reason, every time somebody gets admitted, it shows up on my screen. I'm not sure why that is, but it's okay. So that's a quote from James Watson. I'm assuming everybody knows who Watson and Crick were. And some of these covers now and books are a little bit dated, 10 years in some cases. But there was a cover of Time magazine, which is, why your DNA is not your destiny. There's a couple of books that have come out since this one. This is a pretty decent one. A whole section of Nature, one of the premier journals in the world, was covering this topic. And of course, there's textbooks that cover it as well. This is just the field of epigenetics, which is what I'm going to be talking to you about. Okay, it's not letting me move. There it goes. So there's lots of different definitions. I like this one by a friend of mine, Art Riggs, and a few other people. The study of mitotically and or meiotically heritable changes in gene function, which cannot be explained by changes in DNA sequence. For those of you who know your Mendel, this means non-Mendelian inheritance. That means you can inherit something from your parents that was not encoded in the DNA that you got from your parents, which is why a lot of biologists love this field, because it's just not your standard genetics. It's epigenetics on top of genetics. This field is driven by several processes that go on in cells. It's DNA methylation, which is the one that we primarily study, but also there's changes in histones, which are proteins in your nucleus that which your DNA is wrapped around. And then there's RNA, which I'm assuming you guys have had some exposure to. And RNA is also one of the three pillars of epigenetics. So those are the three mechanisms that can lead to epigenetics. And we study actually this process in bacteria. Before we got started, I mentioned that we had studied some of the bacteria that cause periodontitis. And what we were studying there was a very similar kind of process as you're going to hear about today. It's just that those are bacteria. They're not eukaryotes. They're not, like humans are eukaryotes. And so what I'm going to be talking today about is this process down in here, or these processes down here, and how we can intervene with these molecules that we've discovered. So that involves, you'll hear a little bit about imprinting, exon activation, host defense, and gene regulation. And I have a couple slides on this, so I'm not going to do too much. This is just a marker for me to remind myself that I'm going to be talking about what's called exon activation in mammals, and it occurs in other organisms as well. But when it occurs in mammals, in females, it leads to this tricolor phenomenon in the calico cat. And of course, women are female mammals, and they also have this phenomenon. It's just not color linked in women, but they are. I have two daughters, and I always kid them that their skin doesn't look like this, but they are actually mosaics themselves. They're just as mosaic as this cat is. And then this is actually a really interesting slide. I don't have the time to get into, but I'll talk about the process in just a second. This has to do with what's called imprinting. And it turns out that the DNA that you get from your father and from your mother, even if it's the exact, like the X chromosome, for example, in women, the two X chromosomes could actually be identical in DNA. They're not, but if they were, they could still be, they would still be different, because the information that comes from the father is different than the imprint coming from the mother. What is that imprint? That imprint is one of the epigenetic processes, the one that we study, which is DNA methylation. And so what's really cool is that you're passing on to your offspring information that's encoded on the chromosomes that's different if you're male than if you're female. And then a normal adult, a normal person, gets imprint from both the father and the mother. And that's the reason why we are normal. Okay, so let's see. Okay, these are now, I really got to update this because it continues on. I don't follow this as much anymore, but those of you who are interested in this, and I do get emails from people, both family members and just people who follow, who are fascinated by epigenetics. There's an amazing number of studies that have shown this. This is definitely not the area that I study, but it's just so fascinating, where people have done now DNA studies on people that have been exposed to something in their life. Women when they're pregnant, or fathers if they smoke, or something, and then they look at the progeny, the offspring, and then the offspring of the offspring. And it turns out that, and some of this is sort of like just, it's like, it's a urban legend kind of stuff. You have a consequence on your progeny, but it's not at the DNA level. It's at it's epigenetic. It's being passed on without actually mutations or genetic modifications in your DNA. There's something else going on, and that's passed on from one generation to the other through one of those three mechanisms I just mentioned: DNA methylation, histone or chromatin changes, or some aspect of your RNA. So it's truly fascinating. I highlighted in red a couple of them. So another researchers at Tufts are studying the genes of animals dependent on opioids to see how epigenetic alterations caused by drug exposure affect the opioid sensitivity of the animal's offspring. And the answer is it does. And so, and it's not because there's mutations, it's because there's some kind of change in how that DNA is read. And I'll explain that in a second. The one down below, male rats whose mothers receive a fat dose of this drug late in their pregnancy are highly likely to be born with defective testicles and reduced fertility. Again, not because there's a mutation in the DNA, it's because of some epigenetic change that got passed on. And what's really amazing is it's passed on multiple generations. Okay, so again, biologists love their model organisms. Humans can't really be studied. Well, they can now because of the sequencing has gotten so cheap. But there's things you can't do with humans that you can't do with animals and model systems. And I just wanted to kind of impress upon you that every one of these examples, there's two different kinds of yeast. Here's an organism that you may not have heard of before. This one has led to several Nobel Prizes. This is the plant version of all these other ones. Corn, super studied. Here's a nematode, and here's the fruit fly, and of course here we are. And it turns out that every one of these has literally hundreds or maybe even thousands of biologists studying those epigenetic processes in those organisms. And it's truly fascinating. It's just another world into itself. I'm not going to go through all these. These are just really cool, different. Each one of these organisms has its own biological process that is pretty highlighted in those organisms. Yeah, I'm not going to go through these, but they're fascinating. Okay, so now let's see. Okay, here's just a very short dive into this thing called dosage compensation. It's the thing that leads to the calico cat I showed you down here, or I showed you in the previous slide. And what it comes down to is that the sex chromosomes, females in the case of mammals have two X chromosomes. Males have an X and a Y, and female mammals are female not because they have two X's, it's because they don't have a Y. And it turns out that any organism that has this kind of sex counting of chromosomes, like mammals, they have to find a way to dose it, because women or females don't need two X chromosomes, they only need one so they have to silence that one of them. And so the silencing process is epigenetic, and it's usually involves DNA methylation. And it's truly fascinating because in the case of the calico cat, that process, and I don't want to go into this little genetic diagram, but that process is color-coded. It's not color-coded in humans, obviously, but because the color coding, you can actually tell that this process is leading to different colors within the fur. Now, what's really fascinating, I don't know if I have it up in here, maybe I have it somewhere else. No, I don't include it here, is that it actually happens early in embryogenesis, early in the development of the embryo. In the case of the cat, it's around 80 cells. At 80 cells, it's basically a blastula, a little ball inside of the mother cat. Those cells make a random decision about which X is going to get inactivated. And from then on, for the rest of the life of that cat or human or woman, those X's are going to be inactivated. It's either the father's X or the mother's X. So the normal process is random. However, I think I have, here's the example I was going to give, Turner's syndrome. There is a syndrome in humans called Turner's, and I didn't spell it right. And it is the result of these girls, they only inherit one X chromosome. So in principle, you could say, well, they could be normal because you only need one X. But it turns out that the imprinting, the marking of which genes get turned on and off that have to do with, sex-related imprints is different for male and female. And if you're a Turner's girl, which there are, it's not a common problem, but it's out there. It's much better to get your X from your father than from your mother, because it turns out that the imprint will lead to a much more normal, human than it would be if you got it from your mother. The logic of that I don't know, but I mean it just happens. Finally, this thing about imprinting is really fascinating. It turns out that there's a great paper now. I still use it in one of my classes. It's now quite old. It was titled "Mickey Has Two Moms," and it turns out that if you followed what I said about imprinting, you could imagine that you could take the imprint from a male, and you could change the DNA of an egg, the DNA in an egg, in this case it's a mouse, so that it has the imprint of a male. And that's what these people did. They then took that DNA, that nucleus basically, and they put that together with another egg of a female who had normal female imprints, and they got offspring. Amazingly, they got viable offspring, meaning that those offspring could have babies themselves. And that's what this title says. Mickey has two moms, meaning you do not need males. This is a really fascinating and big topic. It has to do with parent-offspring conflict. The mother is in conflict with the gestating offspring for resources, and the male wants his offspring to be big, the female wants to live another day. And so imprinting and control of that process is kind of the result of both imprints having sway during that early embryogenesis process. It's really a fascinating field, but again, I'm not a biologist. I just wanted to give you some background on what we study here. I'm not going to talk about this, but this has epigenetics is incredibly important for any kind of cloning of either farm animals, and I'm sure it'll happen with humans. But that cloning process, if you could imagine, is the best one would be to think about like if you guys remember Dolly, if you take a somatic cell from an adult mammal, and you take embryonic stem cells, what you can do is you can take the nucleus out of this cell, put the DNA in, or the nucleus in from this cell, put it in here, this embryonic stem, or this stem cell, put it in here, and that can then lead to an adult. And the epigenetics of that process is the most poorly understood, and that's why it's still a very big problem in being able to pull this off. Dolly died prematurely. There are a lot of problems with it. It's very inefficient to do it that way, but it's definitely something a lot of people around the world are working on. Okay, I'm not going to talk about this. This is just more of the same. Okay, getting a little more into what we work on. Just to remind everybody about their high school biology, genetics. Most of us know it can lead to mutations from either environmental assault, something that you do, or too many airplane rides, whatever it is, whatever causing these mutations to happen. If they happen in the germline, then that's going to get inherited. If it happens in your somatic cells, that can lead to disease like cancer, that kind of thing. Epigenetics is really about the process whereby you're not changing the DNA sequence, but you're basically looking, or it's the process of reading that DNA. Some people think of this as like the hardware. This is the software. The idea is that you're passing on changes in how that DNA is read, either at the level of DNA methylation. Here's a little methyl group on the DNA, or little add-ons to, this is supposed to be nucleosomes or histones, and there's modifications. These modifications get passed on. There's also some little RNAs that come along for the ride, and this all gets passed on to the progeny, onto the cells, to the daughter cells, etc. And that's epigenetics. Okay, I'm going to stop. I don't know if there's any questions. Because I talk pretty fast. I don't know if people want to. I can wait for a few, 30 seconds or something like that. And if anybody has a question about what I've gone over so far, I'm going to go deeper and deeper now into the biochemistry and what we do. Okay.
Norbert, I have a question.
Go for it, Mary.
Okay, yeah, not that this process is well enough understood, but from what I understand, there are like 10% of the population that are born with more than 2 sex chromosomes. Some of them
I
are
didn't know it was that high, but I know that it does happen. Yes. Uh-huh.
Yeah, so is this anywhere near related to what's happening in epigenetics, or is that's just totally other?
Yes and no. I mean, I think it's called trisomy. I mean,
Yeah.
so I think what's going on. So the cause of that is, biology isn't perfect.
Oh,
So
yeah.
when you have these divisions, things can happen. And if you have enough divisions, I'm kind of blown away by the 10% number.
Maybe
the
it's
fact-
2%. I don't know. It's some real percentage.
Oh, I'm sure it is. I'm sure it is.
Less,
No, I know
more
people
than
who-
one.
Right. I mean, I know people who study that in mice, and it's an interesting thing because they actually induce it. But the point is that, so the answer to your question is actually kind of interesting. How did it happen? No, that's not epigenetic. But how do you deal with it? How does your body, I mean, how does something like that get through? And I'm sure most of the audience, most of the people here know that women, well, mammals, they spontaneously miscarriage a very high percentage. I don't know what it is. It's more than 10, less than 50, but it's a very high percent, and they don't even know it most of the time. And so that's biology working and saying this shouldn't go through. This should not go through. And so what's amazing is when something can get through, because there can be dire consequences evolutionarily, right? To get something to go through that can pass on that information. So the how you got the trisomy, I don't know, but the epigenetics would be all about regulating the how much of that do you want to read? I mean, it would be confusing if it was like XXY, let's say, or something like that. Actually, isn't that the cause of the those famous Russian women that were super athletic and all that? Isn't that part of that or not? Maybe
No,
yeah.
that's part of it, but there are many women who have testes. The testes just haven't been, they don't necessarily, you don't see them externally.
I see. So I would argue that there's an epigenetic control that would try to modify that, would try to regulate that, because there is a biological drive to have only dosing come from one X chromosome.
Yeah.
So I think the epigenetic, the software, I don't want to imbue it with some kind of design aspect, but the point is that, to the extent possible, there would be some type of, let's bring down the. You wouldn't want to have all the genes from all 3 chromosomes being expressed, which is not normal anyways.
Right.
So I think the epigenetics could be involved in how is it manifested, how is it read, how is it interpreted, but not in the how did you get it in the first place? That I think is just an accident of cell division.
Okay, thank you.
Yeah, sure. Yeah, I haven't thought about that one for a while. Okay, so that's this. This just reminds me to remind you, I've spoken about that DNA methylation, which is a process where this is the way a molecular biologist thinks of your genome. A box is a gene. These are little spots that can be modified by the enzyme on the DNA, can be methylated. I'll show you what that does in just a second. But these are spots in what's called the promoter, which is basically a region in front of the gene that says turn on or don't turn on. If the promoter is methylated, you do not get transcription. You do not get the gene turned on. If it's not methylated, then the gene is turned on. The classic example is globin in your brain. You don't make hemoglobin in your brain. You do make hemoglobin in other parts of your body. And if you look at the globin gene in your brain, it looks like this. If you look at it in those expressing tissues, it looks like this. Same gene. So this is the really fascinating part of epigenetics. This is not transgenerational inheritance. This is about within your own body, your 200 to 300 different cell types. Why are they different cell types? How are they different cell types? It's because of this. Not only because of this, but partly because of this. That means that there's, it's basically a control. It's a control process. So where you don't want to have that gene turned on or that protein made, you do this. If you want the gene made, you do this. 50% of your genes are the same, are turned on in all of your cells, because those are called housekeeping genes. But what makes a liver cell, and a brain cell, et cetera, is that other set of cells which have to be turned on very selectively in time and space. That's controlled by DNA methylation. Histone modifications do the same thing, but they don't modify the DNA. They modify the proteins that the DNA in your chromosomes is wrapped around. I showed you an image before. And then RNA is just obviously DNA leads to RNA, but RNA is information itself. It can actually control the epigenetic process as well. And the classic example is what I've been talking about, this X inactivation. The X inactivation is actually started by one RNA molecule that binds to this I stands for inactive. So every woman on this call has in her cells an X chromosome that's inactive, and it's covered with RNA, and the other one is not. And that RNA process happened when you were 80 cells, 100 cells, 200 cells inside your mother, and it's been passed on for all your cell divisions forevermore. So that's kind of a fascinating process in itself. The only reason I want to show you this is because it shows something that's really important for the rest of the talk, and that is that we have something. This has always been controversial, but it's kind of coalescing around this number. We have some number of genes in the human genome, and these can be either embryonic stem cells. It can be just stem cells, but eventually there's a commitment to becoming something, one of the cell types. Once you go forward, then you can keep on going. Eventually, you lead to these different cell types. The cool thing about epigenetics is that we can do reprogramming, or what happens in your body under normal conditions, under certain circumstances, you can actually go back. And this is the driving force behind a lot of either industry interest or academic interest. How much of this can we control going back? Every epigenetic process has a forward arrow and a reverse. Genetic changes, not so much. If you do a mutation, you're only going one way. So that's the fascination in the field and in industry therapeutics is, can we intelligently reverse this process when it goes wrong? So that's part of that. This is just for fun. I threw the slide in because sometimes people don't appreciate what makes us similar or different from other organisms. And what this shows you is all those kind of model organisms I mentioned before, the two yeast, the plant, fruit fly, mouse, and humans. And what it shows you is that in the yeast, these are all eukaryotes, they all have a nucleus. In yeast, this is the percent of the genome that codes for proteins or for RNA. In the case of, here's another one, but it's a little bit less. Now you go to the plant and it's less. Now you go to Drosophila, it's less. You get to the mouse and the human, and a very small part of our genome actually codes for who you are. There's a whole bunch of your genome that's non-coding, and then there's a whole bunch of your genome that's repeat. Basically, what these repeats are is our primordial ancestors were infected by a virus, a retrovirus, and it integrated its DNA into our DNA. And every time we have sex and we have progeny, we are passing on. We're copying all those viral sequences as well and passing those on to our progeny. So this is us right here. This is what defines who you are, that little itty bitty piece up there. It's interesting. Okay, so every field has its big questions. I don't have the time to go into these, but it's really, epigenetics is a very, very hot area in the world of biochemistry and genetics and biology. It has huge implications for everything like aging, regeneration, stem cells, it just goes on and on and on. It's a really fascinating field. Okay, here we go. This is coming from a high school text. Your chromosome, you spool it out, you eventually get to a nucleosome, which is basically a protein core, and the DNA is wrapped around it. And this is a way to compact your 2 meters of DNA into a cell that's 30 microns wide. I mean, that's an impressive packaging feat. And what's really amazing is that has to constantly be read. So think of it like a book that's totally tightly packed, and pages of it have to be read constantly so that you can make that liver cell, that brain cell, or whatever it is. So how do you get access to those pages when this thing is all packed in like this? It's a super interesting, broader question. So the enzymes that we study are called DNA methyl transferases, DNMTs. This is kind of a chemist view of DNA. You have a cytosine, and it gets methylated at the C5 position with a cofactor that brings in a methyl sulfonium, which you probably can barely see. This is an activated methyl group. That methyl group gets popped on over to here, and you create this product. This is now thioether, and you have a methyl group on the cytosine. So you have 5-methylcytosine. This is that signal that I was telling you earlier that shuts down genes, the gene transcription. That one little itty bitty methyl group. That's it. Pretty amazing. So one way to think about where I'm going with this in terms of the work that we're doing is that this control of which genes get turned on and off, two kinds of genes that are super critical for you to understand is a class of genes called oncogenes. And these are genes that are normally off. They have a function, but they're normally off, and that's why this methyl group is on. They're supposed to be the promoter is methylated, so the gene does not transcribe. It's not being converted into a protein. And then you have a set of genes called tumor suppressors. Both of these are not singular genes; they're many genes. Under normal conditions, this tumor suppressor does what the name implies: is that it suppresses, rapid growth. And just to be clear, most cancer really comes down to two characteristics: immortality and rapid growth. The cancer cell does not play well with others. It doesn't behave according to the kind of the rules of the village. So once it starts to divide, it'll just keep on dividing, and it's immortal. So therefore, it basically wins. And so that is a result of this phenomenon right here. When you get a tumor, what goes on is that you get the oncogene gets turned on. This is not the only thing that happens, but it's a major thing that happens. And that can be either because there's a mutation in the oncogene, or the promoter that controls the oncogene is no longer methylated and this gene gets turned on. The tumor suppressor, in contrast, in cancer gets either mutated or it gets methylated and it gets suppressed. Now, there's a really little simple example of this that I'm sure some of you might be, you might know about this. So familial breast cancer comes from a mutation or a set of mutations in the BRCA gene, and then that is inherited from, in women from their mother, and it's almost guaranteed that woman, that youngish woman, 30s, 40s, will get breast cancer. But those women that get breast cancer, familial breast cancer, are a very small percentage of the women who do get breast cancer. The majority of women who get breast cancer happens 20, 30, 40, 50 years later. And that doesn't come from that mutation. It comes from this phenomenon here. It's because the BRCA gene is now not controlled epigenetically correctly, so that you're not getting the mutation necessarily that gives you that breast cancer. It's because the control of the gene is no longer correct. And that's fascinating, because that means that you can possibly reverse it, which is one of the kind of the major parts of my story today. So there is a drug that's off patent. It's been around for 50 years. It's called 5-AZA. Well, this is only one of them. This is not the one I usually think about. This is called cebularine. This is a young version, a more recent version of the same drug. They inhibit all DNA methyltransferases, and I'll show you that there's more than one of these enzymes. And what they result in, these are the methyl groups on the DNA. If you give this to a patient, their DNA becomes no longer methylated everywhere. Not just in some place that you want it to no longer get methylated, but everywhere it gets unmethylated. And as you can imagine, if you know anything about chemotherapy, this is one of the causes of the problem, is that it's not very specific. There's other kinds of inhibitors that people use in combination with these. They're called Hdac inhibitors, and what they do is they affect the ability of the histones to be modified, these little tails coming off here. And when those tails can't get modified, and you have this process and this process together, you can turn a gene on that was off. This gene was off, now it's on, okay? So that's what you want. All right, so here's the players. This is how a biochemist looks at this kind of thing. Biochemists basically, by definition, we study enzymes, we study things inside of cells. And so the major proteins that are involved in what I'm going to talk about, they're called DNMTs. They're DNMT3A, DNMT1, there's a B, there's a 3L. And the way we write those down is like this. This is the N terminus of the protein. This is the C terminus of the protein. You don't have a scale here, but this is about 1,500 amino acids. This is around 700 or 800, et cetera, et cetera. The reaction that they catalyze is down here. There's an active site nucleophile. You attack here. that activates this C5 for nucleophilic attack onto that cofactor I showed you earlier. You get a methyl group transferred, and now you get a beta elimination, and you get this regenerated, or you get the enzyme regenerated, and now you put a methyl group on here, and that's the methyl group I've been talking about. Most of my lab focuses on enzymes that have, or proteins or processes that are very well structurally characterized. This is an X-ray structure. Obviously, it's color enhanced, but it shows you something that's really intriguing. It shows you that this protein is a tetramer. This is one protein, this is another protein, another one, and another one. This thing can form what it's called a homotetramer, where all four of these are the same DNMT3A. This is 3A, this is 3A, this is now 3L and 3L, but this kind of opens up where we're going with this, and that is that these other proteins on the outside can be replaced so that you get different kinds of homo or heterotetramer depending on which tissue and depending on which cancer. And these proteins on the outside control what this enzyme does. Here's the DNA. This enzyme is methylating the DNA here. And this shows you this little red section up here, or these words say that there's different interactions that we now know about that control this enzyme. This protein interacts with histones, interacts with other proteins, and it's not shown here, but it also interacts with non-coding RNA. So this is a very busy protein, and all those interactions control what this protein does in normal biology. And anytime you have something that controls what genes get turned on and off in biology, it can lead to disease because it can go wrong. Okay, so this project started about 10 years ago, and I had a very enterprising grad student, Celeste Holtz-Schiedinger, who ended up working for Impossible Foods. Those of you who know about Impossible Foods, she went from studying cancer biology and biochemistry to engineering non-meat meat. Anyway, she was studying this, and she was studying this enzyme, but what she wanted to know was, does this protein have to be a tetramer? And the way that she did it is she used a program that calculates the energetics of the interface. These are amino acids right at the interface here, and she did the same thing here, and she did the same thing here. This is that same DNMT3A, but it's the heterotetramer, 3L, 3L, 3A, 3A. What she wanted to do was ask the question, does it have to be this structure? Can it act as a monomer? Why? Because almost every methyltransferase that we had ever studied was a monomer, meaning it acted by itself. It didn't need to be partnered up. So why did biology decide this one needs to be partnered up? So the people who got the structure told us at the time, well, that's because it wouldn't be functional if it wasn't a tetramer. Okay, so she made the mutations based on this Rosetta algorithm, which this is a heat map. It just tells you which interactions are really important at this interface for stabilizing that structure. And what she found was super surprising. What she found was that the crystallographer was wrong. The enzyme, well, partly wrong. The enzyme does not need to be a tetramer. It can act as either a monomer or a dimer. But what Celeste found was that the one function that it needs to be a tetramer for is what's called processive catalysis. And it's kind of cartooned here. I hope you can see it. Basically, you know what? See, I don't think I actually have the normal. Oh, here it is, homotetramer. So this is the tetramer. These little balls are supposed to be the tetrameric protein. The enzyme binds DNA, gets on. The lollipop is a methyl group. It keeps on going, puts more lollipops down, keeps on going, puts more lollipops down until it leaves. This can happen 50 to 100 times. And this happens in your body all the time, or it happens in stem cells in your body, which is your bone marrow, which is again where I'm going with this. Now, it turns out that when she disrupted these structures by making amino acid substitutions and getting a monomer that's active and a dimer that's active, what she found was this: the enzyme could do the methylation perfectly normally, puts a lollipop down, puts the methyl group here, but then it leaves. It doesn't continue on. That means it's very, very inefficient because it doesn't lead to this super efficient, repeat modification. Now, what was amazing is at the time that we published this paper, there was a paper that came out in the New England Journal of Medicine that said that these same exact mutations that Celeste was making were found in AML patients, leukemia patients. And it turns out that these patients, the reason that they get leukemia is because this gene, this DNMT3A, is mutated and it no longer functions the way it's supposed to function. It's doing this not this. That got a lot of blood people excited, and we were contacted by them, and we set up collaborations with them. One of them is Peggy Godell, who's at MD Anderson in Houston, and that's where this project has gone. Okay, so this is just a map of, coming from, literally thousands of patients now. This is the DNMT3A gene. These are the mutations that occur in these patients. The most common one is this, what's called 882, but there's mutations all the way out there. And we studied a lot of these mutations. And what we found, well, before I get into that, I wanted to remind you guys a little bit about, most of us don't think of adults like us having stem cells, but we do. We have stem cells, for example, in our bone marrow. And the way you have to think about it is sort of like, embryonic stem cells are completely capable of becoming a human. They have that capacity. But the stem cells, once you get into differentiation, once you go through, the example I gave earlier of a blastula, for example, what happens is that there's commitment that, you get cells that are going to become skin cells. There are going to be cells that become neurons. There are going to be cells that become different things. Blood is special because we have bone marrow cells. They're stem cells, but they're not as potent as the embryonic stem cells. And so what that means is they can become all the different kinds of blood cells, and there's many more than this. And so what happens is that this differentiation step comes from the DNMT3A. So you and I don't have DNMT3A expressed in any of our tissues except for stem cells. That's important for a cancer therapy, as you'll see in just a second. We have another enzyme that maintains the pattern of methylation. That's this other enzyme I talked about in the beginning very briefly, DNMT1. But the DNMT3A only occurred early in embryogenesis, when you're differentiating the cell types, when you're going from a fertilized egg to all those 200 or 300 different cell types. And it persists in your bone marrow cells, and unfortunately, it gets turned on and mutated in cancer. Somebody just raised their hand. Wynette, what's up?
Sorry, no, I'm not sure how I did that.
Okay, that's all right. At least we know that it works. Okay, so this is the process. This is normal biology. And then this is what happens when things go wrong. So this is hematopoietic stem cells. These are the bone marrow cells, and there's a constant renewal. If you get mutations in 3A, like the ones I just mentioned up in here, eventually what happens is you get you don't get the differentiation, but you get an expansion of either precursor cells or some kind of, it's sort of like just a wayside on the side. The cells do not continue on. There's different kinds of leukemia, obviously, but one of those would be an accumulation of certain kinds of cells. And so this is what one of the causes of early stage leukemia. And this kind of goes back to this idea here. I don't want to dwell on that, this analogy I said, where you have a balance normally, but then what happens is that if there's an imbalance, you get, you're increasing the expression of the oncogenes and a decrease in expression of the tumor suppressor genes. Okay, this is a nice little object that the student who did this project, Jonathan Sandoval, who's now doing a postdoc up at the medical school up in San Francisco at UCSF. And it shows that what he studied while he was in my lab, he just went up there. He studied DNA methylation I've been talking about, regulatory proteins that control this, histone tails, which get modified, and they control this, and small coding RNA. But where he actually went with this was to study small molecules. And that's what he ended up in the last year. And that's what the story was really supposed to end with. So what he did was he was looking for ways to improve. Well, there were two drivers. One is that the currently used drug I mentioned, decitabine, there's another one I mentioned. These are extremely toxic. The therapeutic window, window is extremely small. What is that? That's the difference between the dose you need to have the effect you want as a physician or as a patient. You want to, if you're taking aspirin, you want to get rid of your headache. And if you're taking cancer therapy, you want to get rid of the tumor. And you compare that to the dose that leads to toxicity. Now, I've never done the experiment, but I would imagine I could literally take, spoonfuls of aspirin, Aleve or whatever, and I might get, have a upset stomach or something, but I'm not going to die. That's a huge therapeutic window because all it takes is 2 of those tablets to get rid of my headache. The cancer therapy is oftentimes the therapeutic window is extremely tight. And that's because of a thing called off-target toxicity. What that means is that you have the effect you want, but you have other effects that you don't want. And those effects limit the dosing that you can give the patient. And so that's why you oftentimes get, bad side effects, because you want to have the effect you want, but there's a consequence to the patient. And so that it's one of the 2 major failures of drug development is off-target problems. Anyway, this enzyme, or the currently used drugs, one of them is docetabine. This is the other one I mentioned before. These are what are called suicide inhibitors. They're basically, the enzyme tries to methylate the DNA. And if this has been incorporated in, this is actually what's called a prodrug. If this gets incorporated into your DNA and you try to methylate this is what happens. The enzyme, this gets incorporated, and then the enzyme tries to come along and methylate it. But then once it gets to this part, it can't methylate it and it gets stuck. It can't leave. It just gets stuck here. And therefore that enzyme is sequestered on the DNA, and that kills the cell. So not only is that bad, but also it hits every methyltransferase in your body. Remember I mentioned you and I today, as you're sitting there, you have an enzyme methylating your DNA. It's called DNMT1. That maintains those patterns of methylation that you inherited from your, like early in embryogenesis. It's propagated forevermore. I mentioned X chromosome inactivation in women. Well, that pattern has to continue. If you inhibit this enzyme, you can't continue the pattern. That means that when a woman's cells divide and the X chromosome is inactive, the methyltransferase is inactivated, then one of her X chromosomes is now, the other one's going to start to become active. There's a lot of other problems. A lot of those viral DNAs are silenced by methylation. So a lot of viral DNA starts to pop up. The argument usually in chemotherapy is that, yeah, but we're still we're trying to help this patient, and those are secondary effects, and we can deal with that. But that's the problem with these drugs, which are clinically used today for leukemia. So what we did is we did what's, and this is done in the drug industry a lot, only they do it in the industrial scale. We screened between 1,000 and 2,000 compounds, small molecules. These are deposited by the drug industry for free for anybody who wants to research them, and by the National Institutes of Health, NIH. So if you want to test some of these compounds against something that you want to go for, they will provide them to you. Now, a big part of the driver of this is like the Gates Foundation and the orphan disease groups. Every drug company has a very small effort where they actually help people do this kind of stuff, where they help them figure out how to screen and test. By screening, what I'm talking about is looking for molecules that have the effect that you want. And we want to inhibit the enzyme, but in a very specific way. And the enzyme I'm talking about is DNMT3A. So this is the punchline of the story I'm telling you about. We discovered these 2 molecules. They act in a completely different way. Ironically, or not ironically, or amazingly, they were deposited by a group in this particular library that we looked at because they were able to treat sleeping sickness. It has nothing to do with what we're using them for. Now, the beauty of that is that they have been tested at least in animals and probably in humans. I have to go back and take a look. What does that mean? That means that the compounds that we just described in our recent paper actually work in humans, and they've been applied to humans, not for leukemia, but they have been applied. And so that's really fascinating, because that means we're way ahead of the process. Okay, so what we did is we came up with a very specific way to identify these compounds. What we wanted was not something that binds to the active site, the place that does the chemistry, the place that does the methylation. That would be right down in here, let's say. We wanted to find something that is on the outside, that it's called allosteric. It's away from the active site, because those sites are not so conserved. If we inhibit a surface part of this protein, or if we bind to the surface of this protein and affect the function of the protein, it's very unlikely that those other enzymes, the DNMT1, for example, or anything else, would be affected. What does that mean? We would only be affecting the DNMT3A which is in only a few places in your body. One, it's in your bone marrow, and also it's in cancer. So therefore we would be very selective in what we would be hitting. And so we came up with a screen using anisotropy. I don't want to go into it, where we were able to discover molecules that disrupt these complexes, not inhibit the enzyme by binding the active site, but bind somewhere out here. And that's what those compounds were. And this is supposed to be a cartoon that shows you how it works. This is now a heterotetramer. This is one protein, another protein. This is the 3A that's doing catalysis. It binds DNA, forms this complex, and then this is just the control. But if we add our compounds, they bind to this and they get rid of these two molecules here. So now this enzyme is no longer able to do what this enzyme was able to do because we've been able to disrupt that complex. And in the case of the cancer, these outside proteins are not appropriate. They're not supposed to be interacting with this DMT3A. So we're finding, for example, P53. This is the most mutated protein in colon cancer. This protein binds to our protein and regulate what it does in cancer cells. Okay, let's see. I think I'm running out of time. Yeah, it's 1 o'clock. I'm going to just finish up with a couple more slides here. This is actually, if those of you who are interested in this kind of thing, this is a FACS sorting. It's a fluorescence activated cell sorting. It's pretty sophisticated. And what you're able to do is this kind of tells you what's going on here. This is a marker of the differentiation of these stem cells, and then normally these are different kinds of cells derived from leukemia patients, and there's a whole bunch more over here. But the point is that these cells are derived, and they're stuck. They do not break through. They do not differentiate. And so what you want is you want to cause this differentiation to occur, and that differentiation is scored by this vertical axis. So this is a control. I don't want to go into the horizontal axis, excuse me, but this is concentrations of our drug. And what you see is that in this regime in here, we're causing the differentiation, which is exactly what you want for leukemia treatment, which is what the currently used clinical drugs do, but they're super toxic. Ours is not. So we haven't seen any level of toxicity. So that doesn't mean it's not toxic, but it just means that we don't have the level of toxicity that's manifested in the clinically used drugs. So where are we going with this? What we're doing is we're making modifications of the molecules. We're working with synthetic chemists who are, this is the parent or one of the parents, and we're making substituent changes so that and this is actually where this molecule binds on the surface of the protein. And what happens is we're hoping that by making these changes, we have effects on both potency, meaning not 5 micromolar, but maybe 50 nanomolar. And the drug industry always likes tighter binding. And then the next thing would be even more specific. And now I told you that we don't see any toxicity, but we have to keep on looking for toxicity. So we're not seeing it yet, but we're trying to improve. Basically, this is a standard approach in the drug industry. It's called SAR, structure activity relationships. You're adding things to it, and you're asking the question, what helps, what doesn't help. And this is actually looking at the active site of the DNMT1. We have the X-ray structures of this enzyme, this enzyme. This is what the pocket where this cofactor binds, this S-adenosylmethionine. This is where it binds. Here's the adenine ring. Here's the sugar. This is the amino acid side chain. This is the surface. This is called a Connolly surface. It's basically taking a water molecule and kind of wherever you have a van der Waals contact, that's where the surface is shown. So this is like just a little water molecule that runs on the inside of that, and this is on the surface of the protein on the outside. And these are just the amino acids shown that are inside there that define those van der Waals surfaces. This is one enzyme. This is the other one. This is the one that we're trying to inhibit. This is the one that we're not trying to inhibit. And so what we've done is we've designed molecules that are, this is a slightly different project, but it's still related. What we're doing is we're designing molecules now that can distinguish between the 3A. We want to inhibit the 3A, but we do not want to inhibit the DNMT1. And so, And so what we've done is we've designed analogs now, and we're synthesizing these to be even more selective. I think that's my last slide because I just saw that. And you guys know where we are. I show this slide when I give talks. I'm going to Israel next month. I'll show this slide, and I'm sure they'll enjoy the fact that we live in such a great place. But that's it for me. I think I'm close to, let me see, what's the time? Well, 4 minutes over. So are there any other questions?
This goes back to the earlier part of your talk, and I was curious, does chromosome inactivation happen with other chromosomes besides the XY pair at the whole chromosome level, or does it happen like with individual genes?
So that's a really neat question. So I usually have a slide that shows what you're asking about. And it turns out that during spermatogenesis, which occurs in males throughout their lives, and in oogenesis, which occurs primarily in women while they're still inside their mother, okay, it turns out that during that process, this is the germline, the germline gets entirely whited out. It's like just all the epigenetic marks get removed, except for the imprints. And there's about 50 to 100 imprinted genes within the oocyte and within the sperm, okay? And they don't get whited out. And then what happens is that then once you get fertilization, there's this wave of differentiation. You know, you get to different cell types. Every time there's another differentiation, some genes get turned down and some genes don't get turned down. And so, you know, you get more and more commitment. Obviously with the bone marrow blood, that goes to a stem cell and hovers there for the rest of your life, and it can become all those other cell types. So the differentiation, this kind of phenomena that you're asking about, this X chromosome phenomena, there's some aspects that are unique to the X chromosome, and that's some aspects that are broader. It is a really amazing thing that we completely white everything out except for those 50 to 100 imprinted genes, which, you know, gets passed on to only the sperm and to the egg. It's a really fascinating part of it.
Is there any indication how those 50 or 100 genes are identified?
Yeah, so it's really,
is there some non-coding DNA or? Well,
that's interesting. That's what I did my sabbatical on when I went to Vienna to work with Denise Barlow, who's a mouse geneticist. But yes and no. I mean, okay, we know what types are imprinted from the female. I kind of hinted at this before. The ones that are imprinted from the mother are growth controlling, and the ones that are imprinted from the father are growth enhancing. There's some other things that are imprinted that occur later on in life, but I'm talking about embryogenesis, the embryo growing inside the mother. And so what happens is and the kind of biological logic is the female, she wants to live for another day, so she doesn't want to have Arnold Schwarzenegger inside of her. She wants to be able to have more babies in the future. Whereas the male, it wants the most dominant, most rigorous, most robust offspring that they could possibly have. So you and I are the progeny of that process that's just perfectly balanced. Now, the really fascinating thing, and this, I usually get a lot of attention when I teach a class on this from the undergrads, is I say, and this is why we have sex. Because if it wasn't for that, females wouldn't need males. And so, because they could just have their own offspring, right? And they would just go ahead without it. So the whole idea behind imprinting is it makes sure that there's a need for the other gamete. Do you understand? And I have to say, whenever you compare organisms that can reproduce sexually versus asexually, sexually always wins. So it's not just a thing to make girls or women, you know, like independent. It's a much better way to have reproduction is to be sexual than asexual. But it's a guarantee. Without that imprinting, you wouldn't need men. You wouldn't need males. You know?
Okay, now we can go back to the questions about the leukemia part.
Anything, I'm happy to.
Yeah.
This is really impressive work, Norbert.
It's a lot of fun. It's a lot of fun.
Yeah, I just don't know where to start asking questions, you know?
Well, actually, one of the things that befuddles us is, so if you look at chemotherapy, it was nasty when I was a grad student, and it still is nasty.
It's still very nasty.
Yeah, I mean, I have a big poster outside my office which shows the 200 biggest money-making drugs in the world, and what they are now 30% of them are biologics. They're antibodies. They're not even small molecules anymore. So the reason that the drug industry likes that is because they're benign. I mean, basically, an antibody is a protein. You make your own antibodies, so you're putting something into a body that your body makes itself. What a wondrously, clever trick. The problem with chemotherapies back when I was a grad student is the toxicity aspect of it. Okay,
Yeah.
and so. And the reason that's a challenge is because, like I said before, a cancer cell is a rogue cell that doesn't play well with others, and it's immortal. So, but that's like saying you want to, here's Mary and here's another version of Mary. I mean, or here's Norbert, here's another. You're trying to do a really subtle distinction there. And so all of the ideas that have been promoted for the last 50, 60 years have been just mostly about growth. Something that grows fast is attacked.
Yes.
The thing about, so epigenetic approaches are better because they're reversible. That's the main thing that makes them exciting. The problem is that if you listen to what I was just saying, we can hit the 3A selectively, but you do have 3A inside your bone marrow. So if we give these inhibitors to somebody who has leukemia, it won't be as toxic as the currently used drugs, but it will affect all of the 3A in all of their bone marrow stem cells. So then the question is that going to be a better therapy? We are now setting up animal trials to see how good they are, but I don't know. I mean, I know it'll be better than the currently used drugs, but because like if you think about it, here's all the cells in your body that would have been affected. Now we're only going to affect these. But is that still bad? I mean, is that still what toxicity manifests itself there? Right. So my holy grail would be to find inhibitors that are selective for the mutated form of the enzyme. That's where I want to go. That's my next big Mount Everest, is to go not just to hit the 3A, but to hit 3A that's been mutated in cancer patients. That would be amazing if you could do that. Yeah. You guys probably get a lot of talks on technology usually, or what's no? Yeah. Well, this is a little different.
A mix. Yeah.
A mix. Yeah.
Yeah. Yeah.
Do you guys have any profs in your group, or former profs, or UCSB professors?
Phil Lubin comes to, when we were in person. Phil Lubin would come to meetings pretty frequently.
Yeah, I know Erica, his daughter. Yeah.
Uh-huh.
He's a physicist, right?
Yeah.
Yeah.
We're really, a very diverse group.
Yeah.
Many other people in this group are are now. Don't know that one can ever be former engineers.
Yeah.
A
lot of people in the infrared fields.
Because that's what's here in Santa Barbara.
Sure.
For research. Yeah.
Well, let me make a plug for my other life, okay? So my other life is I started a outreach about 12 years ago with a lecturer here at UCSB. Her name is Darby Feldman. It's called SciTrek. We go through elementary all the way, now we're going into high school. A year ago, I got a $3 million grant and to expand the program. Last year, we went to 3,000 Goleta/Santa Barbara students, and we do it with 300 to 500 university students, and we go into classrooms and we show them how they can do their own experiments. They fail, then they do another experiment and they fail, then they do another experiment and they probably are successful, and then they do a poster. So they basically do science. It's really cool, and we do a lot of assessments, and we published our papers on how good we are. And that grant now is allowing me to set a program up at City College, Channel Islands, and San Luis Obispo. Because the one thing about education that kind of is bad, unlike in science and engineering, if you have a new mousetrap or a new way to amplify DNA, the world will just do it because they just do it. But in education, I don't know, we've all had our favorite high school math teacher, right? But in reality, it doesn't propagate. It doesn't get out very far. And so I'm convinced that if we have a better mousetrap and we can show it works at San Luis Obispo, then the world will listen, I hope. And so anyway, how is this related to you guys? Well, you know, quite honestly, if you know anybody who wants to be part of what we do, what are we looking for? We're looking for people who have skills other than Norbert's skills. So right now I'm hiring staff, 3 staff, okay, which, as you guys know from COVID, that's a pain in the butt. Okay, so I'm hiring staff, a manager, coordinators, people to help me go out. If you know teachers, if you know, some of you might have either, I know, kids or grandkids, somebody that's going to schools, and you know, you talk to the teachers. We usually don't have a problem, to be honest with you, in getting teachers and schools excited, particularly at the elementary level, because those teachers, they have English degrees and they don't teach science. High school, they think they know how to do science, but that's another contentious point. Okay, but I'm just saying that if so, what I don't want to just talk to you guys about this, about you know, crowing about how good it is. I'm proud of it, but actually I'm looking for help. And what kind of help can you guys do? Well, I'm not looking for money. $3 million is a lot of money, but I am looking for help in whatever ways you guys might think about. If you know somebody who's interested in this, if you know somebody how to do that does, well, you guys probably don't, but social media, you know, whatever, you know, help in some capacity to help the program. We can pay or not pay, you know, whatever, you know, so it's just SciTrek, SciTrek. If you look up SciTrek, you'll get us. Okay, so
Great. I just wanted to echo that I used to be, at one point I was on the Goleta School Board, and we were really pleased with that. I did a term of that, and I got a demo, and I was really impressed with how the SciTrek program showed the students by going through the need for having a controlled experiment.
oh yeah,
And changing only one variable. It's hard to think, you know, it's in a textbook and they don't understand, but you want them to have something like, why did that happen? Because you changed 2 things. You need to change one thing. That's just a great concept.
yeah. But you know what's so interesting about your point was 2 things there. One is we all have the scientific method as junior high school students. I guarantee you guys had it. I know I had it. And I can also guarantee you that even my PhD, first year PhD students, unless they've done research in a lab somewhere, they don't really get it. They can give you the words for it. But when they design experiments, I'm like, well, did you do the control? Oh, well, I did it a year ago, Norbert. I did it a year ago. I go, that's not a control, man. You know, control is like right there with the same material, man. Okay, so number one, that's a often abused concept. That's, you only can get it if you experience it. You have to have that failure of not having the control and realizing you're clueless. You have no idea why it didn't work. That's number one. The other one is I do struggle though. I'm the one that's pushing it into the high school now. This summer we brought in 7 high school teachers, but I can tell you these guys, they all feel so beleaguered about, you know, they don't have the time. They, you know, they're studying for the AP exam, which I think is incredibly a mistake, because that is just regurgitation for all. Now, next generation science standards, yes, they all get that. But the reality is that it's hard for me to break in. So if any of you can help me break into local high school science and math now, we're launching a math module with some of the Dos Pueblos and San Marcos High School math teachers. But it's hard because they all think, oh yeah, come in after school. And I don't want to hit the special kids. I don't want to hit the kids that are going to go on and get PhDs. I want to hit the 90% that when they're 25 years old, they know that it's a stupid idea to inject yourself with Lysol. You know, I mean, come on, man. I mean, you know, that just doesn't make any sense, you know, I mean, so if you guys, yeah, I mean, it's a bit frustrating because I think we have such a good thing. And in the elementary, the teachers love it. And the high school and junior high school, we'll get there. I just, you know, I always tell people, I don't believe in reincarnation. It's got to happen in my lifetime, man. You know? Okay, it's been fun talking with you guys. Really has.
Okay, thanks so much.
Yeah.
Really
Okay.
appreciate it.
Link in the chat.
Okay, thanks for that.
Take care.